Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were learning about genomic imprinting, in which we learned about the KCNQ1 locus as well as the H19 IGF2 cluster, and we saw that how both of them are responsible for a disease known as Beckwith-Wiedemann syndrome, how it is a developmental disorder in which the fetal and postnatal overgrowth is seen due to the one or more reasons, and that is also genetic as well as an epigenetic disorder. So it is an imprinted disorder related to imprinted genes. Today we are going to learn about two such syndromes which are also imprinted disorders known as Prader-Willi syndrome, which is abbreviated as PWS, and the Angelman syndrome, which is abbreviated as AS. Okay. So, the imprinted region which is responsible for both of the syndrome that is present on the chromosome 15, okay, and the cluster, the imprint control regions which are involved in those syndromes, they are present besides each other on the chromosome 15, and they are named after those syndromes. Okay, so there are two clusters which are involved known as the AS cluster, the Angelman syndrome cluster, and the PWS cluster. Okay, so the AS cluster as well as the PWS cluster, both of them consist of the genes as well as some regulatory elements which are responsible for those syndromes. So the AS cluster consists of the genes and the regulatory elements which are responsible for, if they are deleted or mutated, they are responsible for Angelman syndrome, while the PWS cluster has some regulatory elements, gene, as well as some non-coding RNA, which are responsible for Prader-Willi syndrome. So that is why they are named after the syndromes. Okay. So the PWS cluster, it has certain genes which are controlled downstream, known as SnRPN gene. Okay. This gene codes for small nuclear riboprotein polypeptide N, which is involved in RNA splicing. Okay, as well as this PWS cluster has downstream the SNO RNA, small nucleolar RNA, as well as the SNRF RNA. Okay, well, the Angelman syndrome, consists of the AS cluster. This consists of the genes as well as the regulatory elements which are involved in AS syndrome, as well as there is a gene downstream to those both of them cluster, which is controlled by the AS cluster, that is known as UBE3A. The ubiquitin ligase 3A. This codes for the ubiquitin ligase. Okay, this is also important gene, because the deletion of this gene or mutation in this gene, this results into Angelman syndrome, and that is controlled by the AS cluster. Okay, and that is why this cluster is named AS, because this cluster controls the gene which, if mutated, results into Angelman syndrome. Okay. So we are going to see how the maternal allele and paternal allele, they have what is the composition of AS and PWS in maternal as well as paternal allele. So the AS helps in methylation of PWS. Okay, so this PWS, this is methylated in maternal allele. So this is the maternal allele. Okay, in the maternal allele, the AS is responsible for methylation of PWS. Okay, while there is downstream UBE3A. Okay, this gene. So now the PWS is silenced. So there will be there will not be any SnRNP RPN expression. There will not be any SNO-RNA expression. Okay, so there will be no expression of SnRPN. Okay, let me write it. RPN as well as SnRNA as well as the SnRNPN, they will not be expressed in this case because the PWS cluster, that is methylated, okay, and that is due to the AS cluster, but there will be expression of UBE3A, okay. So this UBE3A, this gene, this is expressed by maternal allele, okay, as well as it is expressed by paternal allele, but in normal cells. So what happens is that in normal cells, the cells other than brain cells, both maternal as well as the paternal copy of UBE3A gene, both of them are active. Active. But in brain cells, okay, in brain, okay, it can only tolerate one copy. So only maternal copy, so only maternally inherited copy of UBE3A gene is active. Okay, this is a very crucial point because this is only in brain cells. In the cells other than brain cells, both of the copies they are active, but in brain cells only one copy is active. Okay, so UBE3A, this is expressed only in the only in maternal allele, but in the paternal allele this is somewhat different. So okay, so what happens in paternal allele? Let's see. So this is the paternal allele. Okay, so in paternal allele, there is no methylation of PWS. Okay, so AS does not do the methylation of PWS. Okay, let me just make it. So let's say that there is no methylation of PWS. So this is not methylated. Okay, so PWS is not, or say, let's say it is unmethylated. Okay, as it is not methylated, it is active, so it is going to produce the SNRP, so it is going to produce SNRPN. Okay, so SNRPN gene is active, it is going to produce the SNRNA, the SnRNA. Okay, so there will be expression of those genes, but there will not be expression of the UBE3A gene. Okay, so there will not be any expression of UBE3 agent. So this will be silenced. Okay. So this is what happens in the brain cells normally. Okay. But in the Angelman syndrome, the UBE3A gene, this is deleted or this is mutated. So there will be no expression of UBE3A gene. Okay. So what? Let's learn about both of the syndrome one by one. I hope the SnRPN cluster that I showed you above, this is also the PWS cluster is also known as an RPN cluster. So those what do those cluster do and how they are regulated? I hope that is clear. Let's do a quick recap. So in the maternal allele, both of the clusters are present besides each other. Okay, and the AS cluster is responsible for methylation of PWS cluster. Okay, so due to AS cluster is active here, there will be expression of UBE3A gene, but PWS is silenced, so there will be no expression of SNRPN gene as well as the SNRNA. But in the paternal allele, PWS is not methylated by Angelman syndrome cluster, the AS cluster. That is why, as AS cluster is not active, there will be no expression of UBE3A gene, but there will be expression of genes which are included in PWS cluster. So there will be expression of SNRPN gene as well as the SNRNA and the SNRPRNA. Okay, so I hope this is clear to you all. Let's learn about what goes wrong in Angelman syndrome, what happens, what causes Angelman syndrome, as well as the Prader-Willi syndrome. So the first is Angelman syndrome. Okay, so this Angelman syndrome, this is due to no expression of UBE3A gene. Okay, so this gene is not expressed by the maternal allele. So by maternal allele. Because only maternal allele is active in brain cells, right? Paternal allele is already silenced. It is not expressing UBE3A gene. But maternal allele in normal condition does the expression of UBE3A, but in the Angelman syndrome, UBE3A is not expressed. So this Angelman syndrome, this results into brain growth retardation. It leads to the microcephaly, the happy disposition. Children are usually happy. Okay, so and they also show pocket-like jerky movements, the pocket-like jerky arm movements, and seizures are also common in those. They are also common symptoms of those of this syndrome, of the Angelman syndrome. Okay, so these are the syndromes. These are the syndromes by which Angelman syndrome is symptoms by which Angelman syndrome is characterized. So this we can say that if maternal allele is not expressing UBE3A gene, we can say that Angelman syndrome, it is maternally transmitted. Okay, so this is a maternally transmitted syndrome in which maternal gene is silent. So let's list down the causes which can lead to which can lead to Angelman syndrome. So now you know that the key is that there should be no expression of UBE3A gene. So the absence of expression of UBE3A gene could be due to, first, there is deletion of the gene. Okay, so first is the genetic cause, the UBE 3A gene is deleted. Okay. The other genetic cause could be that UBE3A gene is mutated. There is some mutation due to which UB3 is not expressed. Okay, the those are the genetic causes. The epigenetic cause can be that the methylation could go wrong, the DNA methylation could go wrong. So let's say that the PWS methylation does not happen. If the PWS is not methylated, it also leads to the silencing of UBE3A gene. So if PWS methylation does not happen, this can lead to the Angelman syndrome. Okay, so these are the genetic causes. Well, here I'm listing down the epigenetic causes. Okay, so other epigenetic cause could be that there is okay, there is parental, sorry, oh. So the other cause could be that there is paternal uniparental disomy. Means both of the copies of the genome, they are paternally inherited, means they are inherited from father. So so if something goes wrong and both of the copies they are paternally imprinted, if both of copies are paternally imprinted, this means that the PWS cluster will have no methylation, okay, in both of the copies of the gene, and that is why UBE3A will not be expressed. Okay, so means these means instead of having a maternal allele and the paternal allele, if cell has both alleles are inherited from father, both of the alleles are paternal alleles, there will be no expression of UBE3A gene, which will lead to the Angelman syndrome. Okay, so I hope the Angelman syndrome is clear to you all. So these are the causes. So the key factor is that if there is no UBE3 expression, it will lead to Angelman syndrome. And the reasons for no expression of UBE3A, there could be some epigenetic reasons as well as, sorry, there could be some genetic reasons as well as some epigenetic reasons. So I've listed down them here. Okay, just like the Angelman syndrome, let's learn about Prader-Willi syndrome. So the Prader-Willi syndrome, known as PWS. Okay, this leads to the low muscle tone, hypotonia. This is called as hypotonia. So the patients, they have small stature. Okay, they also show hyperphagia. They also show hypogonadism. There is obsessive compulsive behavior which leads to overeating. So that is why there is obesity, or obesity seen in children. Okay, what happens is that in childhood they have sucking problems, so that they can't take nutrition, so they can't drink milk properly. But as they grow, there is some obsessive compulsive behavior is seen in those children, which leads to overeating, which in turns lead to obesity in them. So they will also have some obesity related problems such as diabetes and all. Okay, so unlike Angelman syndrome, the Prader-Willi syndrome is it is paternally inherited, paternally transmitted. Okay, the key to remember is that Prader-Willi syndrome has a P in it, so it is paternally transmitted, or paternally inherited. Well, in the Angelman syndrome, there is M, so you can remember that it is maternally inherited. Okay, so this is just a small trick to remember that. So Prader-Willi syndrome is paternally inherited. So it happens due to the no expression of the genes which are involved in PWS cluster. So there will be no expression of SnRPN gene. There will be no expression of specifically the SnRNA. Okay, there's a SnRD. Okay, let me write. There is a SnRD. I don't remember 115 or 116. I'll comment it down. Okay, so there's this snow RNA which is not expressed, which leads to the Prader-Willi syndrome. So if there is no expression of SnRPN or snow RNA, if they are not expressed, that will lead to the PWS. This means there could be a lot of reasons which can lead to the silencing of those genes. So there could be some genetic reasons as well as epigenetic reasons. The genetic reasons could be that there is PWS deletion. So the genes which are going to code for, means the genes which are going to code for SnRPN or the snow RNA, they are deleted. Okay, so let's say that there is a loss of those genes, or there could be mutation in those genes. Okay, so the mutation could also lead to the no expression, and that could lead to Prader-Willi syndrome. So those are the genetic regions. Okay, but the epigenetic reasons could be that first, there is unimaternal disomy, or let's say the maternal uniparental disomy. Means both of the copies, they are inherited from mother. So both of the chromosome, both of the alleles, they are maternal alleles. So what happens is that if both of the alleles are maternal alleles, okay, if both of them are maternal alleles, there is no paternal allele. Okay, so in this case there will be only UBE3A expression, and there will be no expression of SnRPN gene as well as the SNO RNA, because the PWS is methylated by AS cluster. Okay, so no copy is going to produce SnRPN, and that is why that will lead to the Prader-Willi syndrome. So the unimaternal or the maternal uniparental disomy is responsible for the Prader-Willi syndrome. Again, there could be the problem that methylation of PWS. This also this could also lead to the Prader-Willi syndrome, because if AS does the methylation of PWS, but if something goes wrong and PWS is methylated, if the PWS is methylated, the genes which are included in those cluster, means the SnRPN gene as well as the SnRNPN, that will be silenced. So the methylation of PWS will silence those genes, or maybe if certain thing goes wrong and if there is mutation in AS such that even the even the mat even the okay, so if let's say that there is some mutation in AS such that the AS is methylating the PWS. Okay, so let's say that there is mutation in AS of on the paternal allele. Okay, so the paternal allele which had AS as well as PWS. So in the maternal allele, AS does the methylation, but in the paternal allele, AS does not do the methylation of PWS. So let's say if there is mutation in AS which leads to the methylation of PWS, so the PWS is methylated in the paternal allele, just like the maternal allele. Okay, so, okay, even the paternal allele shows the PWS methylation. This will lead to silencing of the SnRPN gene as well as the SnRNA. Okay, so the mutation in AS could also result, but mutation which is going to lead to methylation of PWS, okay, not like mutation in AS such that the UBE3 is not expressed. It will lead to Angelman syndrome. But if there is a mutation which is going to methylate, which is going to methylate, which is going to cause the methylation of PWS, that is going to result into the Prader-Willi syndrome. So I hope with this everything is clear to you all, how these PWS as well as the AS cluster, they result into Angelman syndrome and PWS syndrome. And I hope with this, let's end this lecture here. In tomorrow's lecture, we will learn about other things such as the polycomb repressive complex as well as the trithorax group of proteins. Till then, see you. Bye bye.